Welcome to the Global Addiction Therapeutics Innovation Day.
Hi, good afternoon, everyone. Very happy to welcome you for this panel that is organized by Hamid Khtiari and team in Innovation Day. Very happy to be here for this panel, and I will co-chair this panel with my colleague Veijiao, and thank you very much, and I will be introducing our first speaker. So Valerie Voon, she's from the University of Cambridge. She will be talking about how neuroimaging can inform new circuit-based addiction therapeutics. And she's professor of neuropsychiatry and neuromodulation at the University of Cambridge. She's a distinguished professor at the Institute of Science and Technology for Brain-Inspired Intelligence at Fudan University in Shanghai, China. And I'm very, very happy to welcome you, Valerie, for this talk. Please inspire us. Thank you very much.
Excellent. Thank you so much for the invitation to speak. It is very much a pleasure to be here and to meet everyone at ISAM. So I'm going to talk about neuroimaging and how it can potentially inform circuit-based neurotherapies. So this is for neuromodulation. We're going to try to focus a little bit on transcranial magnetic stimulation. We are going to focus a little bit more on the frontopolar cortex, the ventromedial prefrontal cortex, and I'll speak more specifically about alcohol misuse. So in this context, we can think about it two different ways. So imaging for the purposes of understanding the mechanistic rationale, or imaging for the purpose of attempting guided precision targeting. So there's slightly different rationales. So when we think about neuromodulation, you're going to hear a lot of this in the next several days. There's different forms of neuromodulation, including invasive, non-invasive, with different forms of stimulation, magnetic, electrical, and ultrasound. And today we're going to talk about transcranial magnetic stimulation. And many of you will be very familiar with this. It's the use of a coil that delivers a magnetic pulse in a time-invariant manner. It results in a magnetic and an electromagnetic field, and then subsequent neuronal activation. And the reason it's of interest to people who are interested in kind of neural networks is that it has greater both spatial and temporal precision in terms of targeting networks. So when you look at this x-axis here, that looks at the effects of different forms of medications or psychotherapy. Medications and psychotherapy have a much more broad effect, more on the macro scale. You can see here neuromodulation has a much more spatial precision effect and also temporally precise effect. And we'll focus in today on TMS. So when we think about the mechanisms that are potentially affected with TMS, it's similar to what people have already been discussing. The role of incentive motivation theories, this kind of shift towards wanting and craving and cue reactivity, the role of negative emotionality, trying to avoid aversive outcomes in aversive emotional states, and then this transition from impulsivity to compulsivity with excessive exposure. And we're really focusing in on the prefrontal cortex, the basal ganglia, anterior insula, and amygdala. So this is a review that was published with NIAAA several years ago, and it went through all of the task-based fMRI studies at the time that focused in on cognitive processes relevant to addictions. And here you can kind of break them down into three main clusters: the idea of incentive salience and cue reactivity. You can see that network that's implicated. The idea of kind of executive and also self-control mechanisms, the kind of negative emotionality component. And then I guess I'm also specifically interested in this kind of shift from goal-directed towards habit control. So this is actually just a slide that kind of reviews the different targets that are used for TMS and also for deep brain stimulation, TMS in green, and in red, the deep brain stimulation. We're going to focus again on TMS, although I do quite a bit of work in DBS. So we're going to focus in on the frontopolar cortex, but actually there's also very good evidence that perhaps targeting the deep anterior insula and more lateral prefrontal cortex, it is FDA approved for nicotine use disorder. And then the lateral prefrontal cortex, again, a target for TMS for depression, but also for addictions. And then, as Hamed had mentioned, cocaine use disorder targeting more the dorsomesial prefrontal, perhaps getting into dorsal cingulate, is CE marked. So one of the rationales for imaging is the question about targeting. Can we use this in terms of precision targeting? We're going to talk a little bit about sort of mechanisms, and then we'll talk a little bit about this precision targeting. But it comes from the depression literature. Right? So depression, TMS was FDA approved for depression in 2008, and it was targeting the left dorsolateral prefrontal cortex and exciting it. So there's different ways you can target that, right? So you can use what's called the 1020 EEG system. You can use more fixed markers, but you can also use MRI like anatomy, or you can use functional connectivity, resting state functional connectivity. And it's a very big area, so it's an important question as to where you end up targeting. So one of the kind of more emerging and compelling pieces of evidence is that of resting state functional connectivity and the hypoconnectivity with the subcallosal cingulate. This is in depression, and the subcallosal cingulate is considered a node for depression, and this is the target for deep brain stimulation. So using the hypoconnectivity between the DLPFC and the subcallosal cingulate, targeting it may improve antidepressant outcomes with TMS. We're still sort of waiting some of the larger randomized controlled trials for this. So what about in terms of substance use? And again, I'm going to focus in on alcohol use disorder. There's very nice meta-analyses, several published. This is just one of them, really highlighting that there's been multiple studies that have been published on TMS for substance use disorders and here alcohol use disorders. And here highlighting an effect on both craving and also on consumption. Okay. The meta-analysis doesn't tell you where it's best to target, because most of the studies have been focusing on lateral prefrontal cortex, left or right, and they're also using either the figure of 8 coil or what's called a HESED or H coils, which in principle are targeting a slightly deeper structure, about 1 to 2 centimeters deeper, but we don't know which one's necessarily more effective based on these meta-analyses. A very nice set of studies have been coming from Colleen Hanlon's group and also from Holly Lysanby's group is really targeting the frontopolar cortex. Okay, so this is using what's called FP1 for the 1020 EEG system. It's just the left frontopolar cortex, and this was run in alcohol dependence, but they've also run it in other substance use disorders. And what they show is that they're inhibiting the frontopolar cortex over a 10-day period, and so using continuous theta burst stimulation. And over the course of follow-up from 1, 2, and 3 months, you see this increase in sobriety with active stimulation compared to sham. They went on to ask, is there a difference in terms of the underlying mechanisms? Here using Q reactivity and fMRI, and showing that if you're in the sham group, there's an increase in functional connectivity when they see alcohol cues, and there's increase from frontopolar cortex to multiple brain regions in the sham group. But with the active TMS group, you see this decrease in functional connectivity to downstream regions. Okay? So potentially a mechanism underlying why this might be effective. So we've also gone on to ask, well, what about other potential mechanisms that might be at play? And here I'll show you a series of studies that we've been looking at using big data. So this is all UK Biobank data. So this is scans in normative controls who also use alcohol. And there's about 25,000 scanned at time point 1, and then 2 to 3 years later followed up in terms of their alcohol use. So we can now separate out what is cross-sectional or state-related versus what is predictive of your tendency towards drinking. Okay? So here in the cross-sectional, the greater alcohol you drink, the more alcohol you use, the lower the volume, so this is anatomical, in the dorsal cingulate. And there's also lower crosstalk in the dorsal cingulate with other brain regions based on resting state functional connectivity. Okay, so that's the dorsal cingulate related to state-related effects. And then we can go on to ask, well, what about predictive effects? What does the brain look like if you then go on to drink 2 to 3 years later? So if you have lower volumes in now the ventromedial prefrontal cortex, okay, just sitting right above your eyeballs, if you have lower volumes and you have lower functional connectivity or nodal degree, you'll go on to drink more. Okay? So this is a region involved in things like representing goals for decisional choices, goal-directed control, for delayed discounting, and there's a range of other kind of cognitive processes that are implicated in the ventromedial prefrontal cortex. But what this is really saying is that there's a difference between state-related correlation versus predictive effects. And so you might want to target different regions depending on what you're after. Another study, so that one was published, this one has just been submitted, and this is again another UK Biobank data set, and now it's focusing on task-based fMRI and using negative emotional reactivity tasks. This is a very standard task. You just show negative images or negative faces and you compare it to neutral, and usually it activates the amygdala. It's sort of this increase in limbic activation. And what we're seeing here is that if you drink more alcohol, you actually have increased activity of the amygdala, but you actually see a very different process with smoking. If you smoke more, you actually have a downregulation or decrease in activity of the amygdala. So this difference is as a function of the substance. And then when you look at top-down regulatory effects, with alcohol, you see this decrease in functional connectivity. This is now effective connectivity, meaning there's a directionality to it because it's DCM. So you have this top-down decrease in connectivity from the ventromedial prefrontal cortex to the amygdala, whereas with smoking more, you have an increase in functional connectivity that's bidirectional from the orbitofrontal cortex. Okay, so substances do differ in terms of brain activity, at least in negative emotional stimuli, and we see differences in terms of the amygdala and the prefrontal amygdala connectivity. Okay, this is a very nice set of studies that comes from the Charité. I've been working within goal-directed and habit control for some time. They've done some really beautiful studies in alcohol misuse. And here again, we're highlighting the ventromedial prefrontal cortex. And what they're seeing is differences between goal-directed control, where you're making choices based on the affective value of the outcome, versus habits, where you're making choices that are automatic based on what was previously rewarded. And this is compelling theory suggesting that we shift from goal-directed towards habitual behaviors as excessive substance use. And what you're seeing here is in the VMPFC, the lower the activity to goal-directed control, the more likely you are to relapse in the longer term with alcohol use disorders. And you see a different pattern emerging with habits. The greater your activity in this area to habits, the more likely you are to drink as a young adult in the future. Okay, so very nice set of findings with goal-directed control and habitual behaviors, again implicating the ventromedial prefrontal cortex. So we've also gone on to ask, this one again is unpublished, does it make a difference if you're targeting regions within the frontopolar cortex? And normally we use the 1020 EEG system, and we target, say, FP2, which is more mesial. AF8 is a little bit more lateral, so it's lateral OFC, and then F8 is much more, probably more ventrolateral prefrontal cortex. Okay? And sometimes people will consider them all together and not necessarily think that they're different. But in fact, if you look at the resting state functional connectivity, we put seeds within these brain regions, and you see a completely different pattern in terms of its connectivity with the ventromedial prefrontal cortex and subcallosal cingulate. So you see with FP2 this increase in functional connectivity, with AF8 a decrease with functional connectivity to the VMPFC, and then you don't really see it with F8. So suggesting that where you target is important. Okay, so just a couple other studies I'm going to just highlight. So this one is using now diffusion tensor imaging, comes from Holly Lisanby's group, and really suggesting that you can potentially target deep structures with anatomical tractography and using that for precision guidance. And this was a study that used interleaved TMS and fMRI. So what they did was they put a seed within the subcallosal cingulate, used tractography to ask where should they target in the frontopolar cortex, and then showed that as you increase TMS, you get this specific activation of the subcallosal cingulate that's now tractography and anatomical white matter tract guided. Okay. And this I should highlight because Hamid is the last author of this one. So this one is using functional connectivity and specifically Q reactivity to ask, can we target functional connectivity based on Q reactivity? This is in methamphetamine dependence, showing methamphetamine cues, looking at both the activity and specifically the functional connectivity between the amygdala and the ventromesial prefrontal cortex, and then showing it clusters mostly on the right ventromesial prefrontal cortex around FP2, and then actually using potentially this functional connectivity may have greater precision than using FP1 or FP2 fixed targets. Okay? Because the electrical field modeling is greater than if you were to model it with FP1 or FP2 fixed targets if you use targeting of the functional connectivity. So I'll just highlight a study that we had been working on. This is published, and we have another dual targeting one that's just going through review right now. But what we're doing is we're targeting two different networks, both the FP1 and the DLPFC, and we do them sequentially. So we inhibit the orbitofrontal cortex, so FP1 initially, and then we excite the left dorsolateral prefrontal cortex, TMS, just this usual standard. And we do this in an accelerated manner, 20 sessions over 5 days. And critically, what you see, this is in depression, what you see is a very rapid improvement in an antidepressant response after 5 days, similar to the same protocol. But what we've done is we've compared it with single stimulation. So it's actually greater clinical response after 5 days compared to just single stimulation in an accelerated manner. And we have some evidence for prediction. So the functional connectivity between the orbitofrontal cortex and the amygdala, the greater the functional connectivity, the more likely you're to respond to dual stimulation. Okay, so I'm just going to highlight where we're headed from here. So I do have a deep brain stimulation study that should be kicking off in the next 2 months. It's been going through 2.5 years of regulatory processes, so I'm extremely familiar with this. We're looking at, again, just sort of balancing between self-control and these impulsive, incentive, negative emotionality aspects. We have a range of different studies that are non-invasive in addition to the DBS study and the TMS studies. And I'll just summarize here. I've been talking to you about sort of the rationale surrounding neuroimaging, particularly for this frontopolar cortex, and then the potential role for precision-guided targeting, potentially using tractography or this targeting the functional connectivity. Okay, and this is my lab. They're amazing, incredibly grateful to them, and thank you again.
Thank you, Valerie, for this amazing talk. You know, it's not my exact specialty, this domain, but I want to ask you, can we expect differences if we do this treatment based on fMRI, but on functional fMRI, for example, having people exposed to several situations that activate craving or other risk of relapse? And is there some add to combine or not? What do you think about that?
Yeah, so I think this is some of the work that Hamed was doing, which is using this kind of, in this case, methamphetamine cues, and then asking about how the brain activates and the crosstalk between the ventromedial prefrontal cortex and the amygdala, and then specifically attempting to target those regions. There is actually a role for what's called priming effects, which is a different construct than what I just described, because that's actually asking about target guidance for TMS, right? But actually what you can do is also, the effects of TMS are state dependent. So meaning it depends on the activity of the network. So there are studies in smoking, for instance, where they show alcohol smoking cues, they activate the network, and then they use TMS and show that actually using those smoking cues are more effective. In that case, you actually don't need fMRI per se, but I guess you could actually then start to ask what brain regions could you optimize that even further.
Achso, ich dachte da. Wo jetzt?
Thank you. Steve Taylor from the U.S., president of ASAM. Just wanted to ask about your findings with respect to alcohol versus smoking and the effect on the crosstalk between the ventromedial prefrontal cortex and the amygdala. Was that difference, the fact that smoking increased it and alcohol decreased that, was that difference something that you expected, or was it a complete surprise when you got the findings? And if it was something that you expected, do you have any speculation of what other drugs like cocaine or which are stimulants versus other CNS depressants like benzodiazepines, what effects those might have?
Yeah, so that's an interesting question. So I'm sort of both a lumper and a splitter. Like I believe, there's certain cognitive processes and perhaps craving or cue reactivity that very clearly have a specific neural network, right? So the same neural network is observed in craving and fairly similar, and is similar to cue reactivity. It's the same network, nucleus accumbens, amygdala, dorsal cingulate, orbitofrontal cortex, so very similar. So I was expecting the same with the negative emotionality, to be perfectly honest. So it's actually fascinating that it is different. They are different effects, and it is conceivable that one might expect that perhaps a CNS depressant or a CNS stimulant might have potentially different effects. But again, this is a negative emotionality specifically, but I would speculate that perhaps maybe the stimulants might be more like smoking perhaps as compared to alcohol. That's a speculation. It's conceivable it might actually have a different downstream effect. Yeah, so sorry, that would have been a completely different talk. So that's a perfect question. So there's various different ways that you can excite or inhibit activity. And the neural activity actually, it has nothing to do with the specific, so it excites or inhibits both excitatory and inhibitory neurons at the same time. But different frequencies will have a different effect. So 10 hertz or 20 hertz will be excitatory in nature. 1 hertz is in principle inhibitory. And then not more recently, probably about 5 or more years ago, they came up with a theta burst design. And theta burst, depending on whether it's this continuous theta burst, that's inhibitory. And then the excitatory one is this intermittent theta burst. So now people have shifted towards ITBS and CTBS because they're shorter. They're much more clinically tractable rather than this kind of 18-minute protocol. We've been using, so I do run this clinically also. I've been using 1 hertz here rather than CTBS because it's too uncomfortable. So I have actually shifted, even though it's 18 minutes long compared to 40 seconds, I still use because it's just more tolerable.
Organized by International Society of Addiction Medicine, sponsors and co-organizers CNRE, Indivir, Spark Biomedical, Salofa, Ethifarm, University of Minnesota, European Union Drugs Agency, UK Office for Life Sciences,